if there's any issues with the sound quality. So I'm currently streaming on TikTok, Instagram, and YouTube at the same time. So there's a high probability that there may be some issues with anything. So do tell me in the comments. If you have any questions, I would recommend going straight to YouTube. The reason being because I'll be able to reply to any questions. So let's get started if there's no issues. In this stream, we're going to be looking at GCSE Biology Paper 1. So looking at the content within GCSE Biology Paper 1, there are 4 key topics. So there's cell biology, organization, which includes things system, the heart, cardiovascular system, infection and response, bioenergetics, which is the small topic. So the way in which I'm going to structure this live stream is I'm going to go through each of the topics and phrase it as if it were exam questions. And this is not just for AQA, this is for all other exam boards as well. So as I'm going through this, I'll be asking it as if it was exam questions. And then together, even you can comment your answers, bullet pointing what you think the answers would be and how to structure them. Because remember, the GCSEs aren't really about learning the content, it's about mastering the exam technique. So let's begin. So let's start with cell biology, which is pretty much the first topic then. The first exam question I would like to ask you is to give 3 differences between the prokaryotic and eukaryotic cells. This will hopefully remind you of the differences between prokaryotic and eukaryotic cells, because a lot of people forget to revise this part of the GCSE. So have a think about it. You can put your answers in the chat. And if not, let's have a go together. Is this live stream suitable for someone who might not be familiar with the topics? Yes, because we're going to be going through them. So instead of just answering the exam questions, I'll try and explain it to the best of my capacity. Okay, so let's begin answering. So firstly, what is prokaryotic cells? What are eukaryotic cells? So we often learn cells as in animal cells and plant cells. So what it's going to be is that eukaryotic cells are basically all cells that contain a nucleus. So their genetic information is enclosed in a nucleus. So things like plant cells, animal cells, etc. Whereas a prokaryotic cell is a much, much smaller cell which does not contain a nucleus. So the way to phrase that in an exam, if you were asked the differences between what is the difference between them, is that a prokaryotic cell is a cell which its genetic information is not enclosed within the DNA. Well done, Cloudy Angel. That's really good. Prokaryotic cells are not animal cells. Eukaryotic cells are animal cells. So in terms of the differences between prokaryotic and eukaryotic cells, one thing that we can say is that in terms of the size difference. So one thing we can say is that prokaryotic cells, or prokaryotes as they're called, are much smaller. So an example of a prokaryotic cell is something like bacteria. And this is obvious, right? Because that's how things like bacteria gets within our system. And that's how we can get ill, because they are so small. Whereas plant and animal cells can't just sneak through our system. We have to purposefully digest them. So clearly prokaryotes are much smaller than eukaryotes. That's one difference we can say. That will gain us 1 mark. This is a 3 marker. Another difference we can say is that prokaryotes contain their genetic material free within the cytoplasm. So, i.e., prokaryotes, genetic information is not enclosed in a nucleus. It's enclosed within the cytoplasm. The cytoplasm is basically the jelly of the cell that makes up the cell. It's not just empty space. Every subcellular structure within a cell is suspended in the cytoplasm. And the final difference we can say is that prokaryotes have something called plasmids. And we haven't touched on that yet, so let's look at that now. Yeah, well done, Anonymous. That's exactly right. Perfect. Well done, everyone, for the contributions as well. That's really good, really active revision. So yeah, everybody is right. They're much smaller. The genetic information is enclosed in a nucleus, and they have plasmids. Now, another key thing that we need to mention is, well, what are plasmids? So prokaryotes still need to enclose their genetic information in something. They can't, even though they don't have a nucleus, they still need to have it somewhere, and that's where they have the plasmids. So a normal cell, you can imagine, looks something like that, and then that's what the nucleus looks like. So it's a plant or animal cell. Whereas a prokaryote, a bacterial cell, will look a lot smaller and often have something called a flagella, which is a tail, and they don't have a nucleus, but they have plasmids, and plasmids are defined as very small rings of DNA, so they look like that. That's what a plasmid is. And only prokaryotes have that, eukaryotes do not have that. Yes, plasmids are small rings, and the cytoplasm is where chemical reactions take place. Really lovely, everyone. Okay, so that was the first exam question, and now you know prokaryotes and eukaryotes. The next topic we are going to look at animal and plant cells, something that you probably are a little bit more familiar with. Now, if you're watching this from TikTok, you'll probably be able to see the exam questions as well. So this exam question, this is a question on here because almost every single student gets this wrong or has errors initially, unless they've done this question before in answering it. So explain why mitochondria in cells are important. So you're basically explaining what the purpose of mitochondria is. Now, think about the question, and I'll tell you what the mistake that almost everyone makes. So people believe that energy is produced in the mitochondria, and that's wrong. Energy cannot be produced because the law of conservation of energy. Energy cannot be created or destroyed. So it's integral. This is often a 2-marker. So the first thing you want to say is that energy is released, not produced. You will lose a mark if you say produced. And why is it released? It's released from the process of aerobic respiration. It's important that you say aerobic respiration because that's the one that requires the oxygen, and anaerobic respiration does not take place there. Yeah, perfect. So it's respiration, but we just have to mention it's aerobic respiration. So explain why mitochondria in cells are important. Energy is released from the process of aerobic respiration. Okay, moving on to the next question. Now, this is a bit more of a difficult question, that's why I'm putting it up here. Ribosomes synthesize proteins. Explain what that means. A lot of people get scared at the word ribosome synthesize proteins, but just think of it all the way from the beginning topic. When you were learning about what ribosomes are, we learned that the definition of ribosomes is that they're where proteins are made. So synthesize, just from deducing this, synthesize must mean where the proteins are made. And then a bit of knowledge from later on in paper 2, this is more of a paper 2 question, but a bit of knowledge. So proteins are made, and then they're made from amino acids. This was actually a 3-marker, so they wanted you to say from amino acids. Therefore, ribosomes are the site of protein production. So yeah, you need to be more specific in terms of the respiration. It literally depends on the marker, because sometimes there'll be more pernickety, sometimes they won't. So yeah, it'll be safer to say the whole thing. So in terms of the ribosome synthesizes proteins, it just means ribosomes are where proteins are made, and then expand on that and say, okay, where are proteins made? How are they made? They're made from amino acids, and therefore ribosomes are the site of protein production. Let's now move on to the next question, which also a lot of students often don't answer this one properly, despite it being looking quite easy. So what is the function of a nucleus? Now, firstly, for this question, make sure you're always looking at the marks. This will normally be a 2-marker. The reason why it will be a 2-marker is because there are actually 2 functions of the nucleus. A lot of people think they're just one. So make sure you're saying that the 2 functions of the nucleus, Firstly, avoid saying it's a powerhouse of the cell, because they know that that's more of a key stage 2 definition. So you want to say, firstly, it controls the activities of the cell. And secondly, you want to know that the nucleus also contains the genetic information. And that's all you have to write for those 2 marks there. So if you thought you would have only put one of them, make sure you're noting down the ones that you would have missed. Again, well done for everybody who's contributing in the TikTok and YouTube and Instagram. Okay, let's move on to the next question. Now, this is just a little bit extra. So, name one type of cell that does not contain a nucleus. So, think about this. What types of cells do not contain a nucleus? Now, for this question, you can list any type of prokaryotic cell, for example, bacteria. You can say a red blood cell. And perhaps you can say, from your plant knowledge, a xylem cell. So those were the 3 most popular options that people came up with. Let's now move on to the next question. Also, if anybody has any questions, do tell me and I will help you guys. If you want me to go through this in a different way, also tell me, because I'm doing this to hopefully help you. So now we're going to go more into specialized cells. So in terms of GCSE Biology, across all the specifications, they want you to be able to know the different types of plant specialized cells and animal specialized cells. So we're first going to review animal specialized cells, like the one here. So this is a 3-mark question, but it could easily be a 6-mark question. Compare sperm cells and nerve cells. So get a piece of paper, bullet point down everything you can think of, and let's look at the answers. So, comparing sperm cells and nerve cells. Firstly, your first mark is looking and comparing the functions. What is the function of a sperm cell? What is the function of a nerve cell? So the function of a sperm cell is to fertilize the egg, or fertilize an egg. The function of a nerve cell is to transmit electrical impulses throughout their central nervous system. So transmit electrical impulses. So that's 1 mark, comparing the functions. Now, the next marks will be comparing the adaptations, because you would have been expected to learn about different adaptations. So you can say a sperm cell has a streamlined shape. A part of the head of the chromosome of the sperm cell is called the achromosome, and the achromosome contains digestive enzymes. And these digestive enzymes break down the cell walls of the eggs that they're fertilizing. So imagine this is a sperm cell. This part here is a chromosome, and it's crammed full of these enzymes that help digest the egg. And then another adaptation that a lot of students say is that it's full of mitochondria, which allows a lot of release of energy. Now let's look at nerve cells. So nerve cells are the ones that people often struggle with. So nerve cells, as we said, send electrical impulses to the brain. So key ideas is that it has a long axon, which just means it has the cell itself is long, it's got a long body. And the longer each cell is, it means that it can easily transmit a longer distance. So that's an advantage. Another thing we can say is that they contain myelin. So myelin is basically the insulating sheath, because these cells can overwork and get really hot. Therefore, the myelin basically helps by cooling it down and keeping the transmissions quite efficient. And that's it. So if you were to just give 2 sperm cell adaptations and 2 nerve cell adaptations, that would be your 2 marks. Perfect. Now, before we go into specialized plant cells, we're going to look at a question about what does differentiation mean? What is differentiation? So think about what differentiation is. Now, this question is a 2-marker. So they expect almost 2 bullet points. And by the way, in most exam boards, guys, you are able to write in bullet points. And I would suggest to do that, because it not only helps you and your brain in terms of the clarity, it also helps the examiner. And if you're making the examiner happy, that's only a good thing. So what does differentiation means? In the mark scheme for AQA, it's defined as how a cell changes over time as the first mark. And then the second mark is to become specialized for a specific function. So differentiation is how a cell changes over time to become specialized for a specific function. So just in terms of your GCSE knowledge about what does that actually mean? Yeah, perfect, guys, putting the answers in, that's really good. So looking at what exactly differentiation means, it's basically when you think about cells just in an embryo, in a ball of cells, that embryo is all just repeated cells, and eventually that embryo is going to become a person just like you and me. And in order for it to become a person just like you or me, the cells need to change. It can't just remain that. And therefore, differentiation is basically the process of those basic cells changing into those very differentiated, specialized functions. So that's just to help with your understanding. Let's look at the next topic now, which is we were going to look more at stem cells, but let me just make sure that we've completed all of the knowledge so far. Okay, so we are going to go back onto microscopy in a minute. We're going to look at required practicals towards the end of the stream. So we're going to go straight on to stem cells now. So start thinking about what are stem cells? And then we're going to have a question. Compare animal and plant stem cells. So firstly, what are stem cells? And this is another question you could easily be asked to define what a stem cell is in a biology exam. So just making sure that you understand. A stem cell is an undifferentiated cell. So it's not been specialized yet, and that would gain you 1 mark. It's an undifferentiated cell, and then the second mark would be that can give rise to any type of cell. So that's not answering this question, but it could be answering a define a stem cell. So that's what a stem cell is basically a cell that can turn into any other type of cell. So just how I was talking about that example, which I seem to have lost. Oh no, it's here. The example of the embryo becoming specialized. All of these tiny cells must be examples of stem cells because they are not specialized yet. So now let's use our knowledge and apply it to the question. So compare animal stem cells and plant stem cells. So in terms of animals, key things to note in terms of animals is that for stem cells, you can have 2 different types of stem cells for animals. You can have embryonic stem cells, which we just discussed, or you can have adult stem cells, also known as bone marrow stem cells, because they exist in the bone marrow. Now, embryonic, it makes sense for stem cells to be there because it's going to give rise to all types of cells. But bone marrow, that might be confusing. Why are there stem cells in the bone marrow? Now, the reason why is because red blood cells, as we saw in a previous question, they don't have nuclei. And if they don't have nuclei, then it's going to be, they're unable to undergo mitosis or meiosis and reproduce. And obviously, we're going to need more blood cells. We don't just stick with the ones we're given at birth. So what happens is you have the bone marrow, and every time you have a stem cell in the bone marrow, imagine that's your stem cell in the bone marrow, what happens is every time it splits, half of it becomes a stem cell, and another half of it becomes a blood cell. And I'm being broad when I say blood cell because a blood cell could mean a white blood cell, a red blood cell, or a plasmid. So any of those things, they can't reproduce themselves, and therefore the stem cells in the bone marrow do it for them. So those are the different types. Well done, everyone in the comments. So yes, a plant stem cell can be a meristem tissue, and that's what they're called. So plant stem cells, on the other hand, are called meristem tissue. And they basically exist on any root or shoot. So on the roots of plants or on the shoots of plants. And the reason why they're on the roots, on the shoots, because on the tips of these, that's where all the growing is taking place. It's trying to reproduce and grow. And that's where plants are able to grow throughout their lifetime. Whereas animals we only mainly grow from the embryonic stage. And then the adult bone marrow stem cells, these are used for just repair or replacement of cells beyond that point. For the people who are asking in the TikTok, this is not pre-recorded, so I can see all the comments as well, so you can feel free to ask. And I hope the audio is well, because normally this is a bit chaotic. So we're now going to go on to the next question, which is explain the advantages and disadvantages of using plant stem cells. Because plant stem cells are used for a variety of purposes. So you're going to need to be aware of firstly, what are the purposes of them? And then secondly, what can they be used for then? The audio is good. Thank you everyone for reaffirming that. So advantages and disadvantages of using plant stem cells. Firstly, what's your understanding of how plant stem cells can be used? So plant stem cells can be used to clone rare species. And the way all of this happens is paper 2 content, so I'm not going to go into it too much, but that could be one mark. And make sure that you're making it clear that this is an advantage. Another advantage of perhaps using plant stem cells is that crops that have advantageous parts can be used, so crops with useful features can be maximized. So those are 2 advantages you can discuss. And in terms of disadvantages that we can mention, for those asking about chemistry and physics, yeah, so we're going to, okay, we can go on to physics tomorrow then. Thank you for your requests. If you have any more requests, people, about what content you want to see, then just put it in the chats and put it in the comments so I might be able to see it for a longer time. So those are advantages. Disadvantages include things like, if we're cloning those rare species, then what could happen is it could, and what would happen is it would increase the competition between the different plants growing. Because if we're cloning all of these species that have really useful properties, it might outcompete the normal wildlife, and that could be really dangerous, because then those could go extinct, and so it's not really good. So it could therefore affect the population of other plants negatively. And then another disadvantage is that, which is the most common one, clone rare species. If we're cloning them, then they're more susceptible to disease. Because if all of the plants are made from the same exact one, and if all of them have the same genetic makeup, and one disease was to wipe them all out, then you have absolutely nothing left. And yes, so I will do physics paper 1 tomorrow, chemistry paper 1 the next day. Thank you guys for your requests. And I will also do English literature for those asking on TikTok. If you are finding this helpful, guys, do tell me. But if you do want me to do something else, like focus more on going through the content rather than exam questions, then also tell me that as well. So now let's go on to microscopy and looking at the wonderful, wonderful required practical. Required practicals are really important and integral. Thank you guys for the comments. The required practicals are really, really important. Almost half of the paper in biology, now I'll tell you guys this, almost half of the paper are practical questions. And what they try to do is they try to slightly tweak the question so that it's not exactly the required practical that you learnt, but it will be using the same method. So don't get thrown off if it's written differently. For example, if you guys remember the osmosis practical that we'll be looking at soon, the way you guys learnt it through the required practical is using potato. But I've seen many times in the exam they've replaced potato with something like carrot, or they've replaced it with something like onion. So don't get thrown off if it's not exactly the way that you've memorized it. Thank you guys for your comments. So let's go on to the microscopy required practical. And firstly, just looking at microscopes as well, because you need to know a little bit about that. So firstly, light versus electron microscope. What's the difference? Basically, a light microscope is more readily available. You can use it in schools because it's much smaller. An electron microscope is much more expensive, therefore it's less available. And in terms of what is actually the difference between them, well, a light microscope, as the name suggests, uses light waves, whereas, yeah, any other vegetable tissue, well done. Yeah, guys, so before the actual exams, I will also be doing live streams just like I did last year, so hopefully that will help you. Yes, so the difference is electrons have a much better resolution. So it's a confusing idea. The higher the resolution of a microscope, that means that the smaller you can see, right? So if it was 1,000 resolution, that means you can zoom in 1,000 times. If it had a low resolution, it means your ability to zoom in to magnify decreases. So electron has a higher resolution. A light microscope therefore has a lower resolution. And therefore you can say that electron microscopes allow greater magnification, whereas light microscopes have limited magnification. Now, a common question actually asked, yes, I do the lives for all of the subjects, because I didn't do my GCSEs too long ago, so I'm still able to go through the content and I have my notes with me. So light versus electron microscope, another key question that they tend to ask is what structures can you see with a light microscope, or what structures that you can see with an electron that you can't see with a light? So with a light microscope, we said that it has lower resolution. So you'll be able to see things like a cell wall and the nucleus. Whereas with an electron microscope, of course, you'll be able to see those things, but you'll be able to see subcellular structures such as mitochondria. That's a good one that they tend to ask about, but you could also say ribosomes as well. That question comes up quite a few times as well. Okay, so moving on to the next question. So we looked at light and electron microscopes. Another key question, actually, let's go through the required practical first. One second, guys, I'm getting a bit of issue with the stream. Let me just sort this out. Thank you guys so much for watching and commenting, by the way. It makes a huge difference. I've been doing these live streams for a long time, so thank you so much for watching. Can I ask how old you are? Guess how old I am. Okay, but whilst you do that, let's move on. So let's actually look at the required practical. So in terms of the required practical, what do you guys need to know? Well, for every single required practical, whether it's biology, chemistry, physics, I recommend you need to be able to bullet point out the method. You need to bullet point out the key mistakes that could be made. And when I mean mistakes, like such as human errors, this error, that error, and just be able to answer the common questions that are asked about them. So microscopy, the key bullet points of what happens. Firstly, let's go through the equipment. What equipment is going to be needed? This one isn't too bad. It's just a light microscope. What else could we say? A glass slide, a cover slip. (21:17). I am 20 years old, guys. Hi, when are you going to do poems? I'm going to do poems in a future live stream. I will organize which live streams I'm talking about soon. Okay. So equipment, light microscope, glass slide, cover slip, scalpel, iodine in order to stain it, and yeah, that's it. So step 1 will be to cut a section of the onion. Oh God, what's going on? Cut a section of the onion. This will be done with the scalpel. I should set up a Discord server. That's a good idea. I'll look into that. Thank you for that. So cut section of onion, scalpel. Second, what you want to do is, because you're not going to just use that massive chunk of onion, so what you're going to do is you're going to then peel the epidermis of the onion. Make sure you're using all of these keywords. Examiners will just go tick, tick, tick. So peel the epidermis of the onion very, very carefully. You can use a scalpel again. And you're then going to place that epidermis onto a glass slide. And a key thing here, make sure you mention this, or they might ask you a question, why might you have to be careful when placing the epidermis on the glass slide? So you want to say, you need to be careful because you don't want to trap air bubbles. So that's why you need to carefully place the epidermis onto the glass slide to not trap any air bubbles. You use iodine so you can stain it on the subcellular structures and visible to see. Perfect explanation there. Well done. So yeah, you place it on there. And so after you place the epidermis onto the glass slide, what are you then going to do? You're then going to stain the specimen, as mentioned in the chat. So stain the specimen using iodine. And what's it going to do? It's going to highlight the subcellular structures for you to be able to see. And then what are you going to do after that? You're then going to soak any excess iodine with a paper towel. Remember, you don't have to put all of these points to get 4 marks in the exam. This is just all the points from the mark scheme. So it's beneficial that if you were to miss one, you have as many as you can so you can bag as many points as possible. Soak any excess, then you're going to place the cover slip on top very carefully again to avoid trapping any air bubbles. And then a lot of people get confused with how to actually use a microscope. So what you're going to do is you're going to start with the lowest objectives. So they tend to be 4 magnification times 4 times 10 and times 40. So you're going to start with 4 times 4, and then you're going to turn the focusing wheel and the focusing wheel is going to physically move the lens closer to the slide. And then you're going to turn slowly adjust the focusing wheel. So here it's going to be quite fast the first time you're doing it, and the second time you're doing it is you're going to slowly adjust the focusing wheel in order to ensure that there's more detail. So until it comes into focus. And that is pretty much it. And then you just want to say repeat for the higher objective lenses. And that's the required practical. A key question that they ask is, why can't you see through the microscope? Why might it be difficult to see? And you can say one of the objective lenses is a bit dirty, or you could say something like, the microscope isn't focused properly. So that's one required practical. All done, really well done. We're now going to look at cell division, things like mitosis. I have a feeling we won't be able to go through the whole of the content within this one live. And I don't know if I'll be able to do till 7 o'clock, but I'll try and do as far as I can. And then don't worry, guys, because I'll just keep posting more lives. And the more you find this interactive, then the better. So, mitosis. Firstly, what is mitosis? What's your understanding of mitosis? And what are the key bullet points? That's the key thing. What are the key bullet points for mitosis? What are the stages? Now, depending on what textbook, there are many different explanations of what are the different stages, but here's one simplified version which you can almost apply to every exam question. By the way, guys, the place I'm getting all of these questions from, I'm basically, if you haven't heard of this before, if you haven't heard me mention it before, I made this massive document with all exam questions for biology, chemistry, and physics, every single exam question in literally GCSE history, and organized it into topics. And that's on my website for free, guys. You can check that out. So it's got all of them there. So that's where I'm getting them from. So mitosis stages. Stage 1, which always try and mention, remember, stage 1 is the longest stage. It's a stage where the chromosomes, the subcellular structures, the DNA, everything is duplicating and increasing in number. Stage 2 is then when one set of chromosomes is pulled to either end of the cell. So imagine you've got your cell and you've got your chromosomes here. So they've duplicated, so you'll get half being pulled to either side. And then stage 3 is the final bit that's being separated, which is the cytoplasm and the cell membrane. So they are then dividing to form two identical daughter cells. So that's the process of mitosis. I memorized this version because it is applicable, but then in some versions there are like specific names for each of the stages. I wouldn't say you have to go and memorize them. I think that's excessive for how much you need to know. But if you feel better memorizing them, then you can do that as well. And also another thing, this is often called the cell cycle as well, because it's basically the way in which cells reproduce, and that's quite integral. And leading into that, a key question that could be asked is, why is it important? Why is mitosis important for multicellular organisms? So let's look at that question. Explain why mitosis is important for multicellular organisms. So here we really need to think, why is mitosis going on? What is the purpose of that? It's similar to what we talked about in terms of the adult stem cells and the bone marrow and what's going on there. The main reason why we have that is for the repair of cells or the replacement of cells. Imagine if we didn't have mitosis, that means if we injured ourselves, the cells wouldn't be able to rejuvenate, and therefore we would literally just keep bleeding or whatever till we died, and we would be very vulnerable. And that's why mitosis is important for repair and replacement. And yeah, that's the main two functions of it. Reproduction, no. Well, yeah, reproduction, it is essential because I'll quickly explain this. This is more paper 2 content. But in 2023, they actually put a question about this in the paper one. So the exam boards were a bit evil in the AQA. So I'm definitely going to try and, if I were you, I would just try and have a good understanding of everything. So in terms of reproduction, what's happening is the sex cells, also known as the gametes, so you've got the sperm cell, and you've got the ovum, the egg cell, right? These are gametes. So what happens is gametes, the way they reproduce, because they already have half of the DNA, they only have, so everything has double the amount of chromosomes, 46 chromosomes, an average cell in the human body that has 46 chromosomes, whereas gametes only have 23 chromosomes. So gametes only have 23 chromosomes, which means they can't reproduce via mitosis. So the way gametes reproduce is via meiosis. And then you get two identical cells, you get genetically different cells. So meiosis is a process where you get genetically different cells. So that's how the gametes reproduce. And then when fertilization happens and these two fuse, you then get an overall cell, which then splits into a ball of cells. So the zygote splits into the embryo, or the embryo, splits into the zygote. So you've got your ball of cells, and the way that this happens is through mitosis. So you're correct in a way when you're saying that mitosis is needed for reproduction, but it's not needed for the reproduction, it's not needed for the fertilization phase, it's needed for the how cells are becoming specialized from a baby. So embryo specialization. But because that's a bit hard to explain, they don't really ask you to explain that. I hope that's clear. If you do have any questions, anyone, then just keep commenting. Phoebe from TikTok, don't worry, you're not going to fail. You can do this. You can understand and hopefully reach that point where you understand it, because just like you, I didn't really enjoy learning any GCSE content, but after just putting the work in year 11, I admit it was very last minute, but after putting the work in, I was able to get there. So you definitely can as well. Okay, so we've looked at mitosis, we looked at why it's important. Let us now move on to looking at things like diffusion, osmosis, and beginning to look at the next required practical. So I think what any GCSE student has always been good at is defining what is meant by the term diffusion. So this is normally a 2-marker. What is meant by the term diffusion? So diffusion is the net movement of particles from an area of higher concentration to an area of lower concentration, going down the concentration gradient. So it's 3 bullet points there, but it's only 2 marks. What grades did I get at GCSE? So I got all 9s, but I definitely wasn't predicted that at the beginning. So it was after a lot of hard work in the final stages, but that's why I believe that anyone else can also achieve those top grades. Am I live? Yes, I am live. So we talked about net movement of particles, high concentration to lower concentration, going down the concentration gradient, diffusion. So when we're talking about definitions, I hope you're just not memorizing it, but you're also understanding what that means. So when we're saying down the concentration gradient, you can look at it as if it looks like this. So initially it's got lots of concentration, but then it kind of fizzles out there. So it's going down that thing, where it's going from an area where it's so close together to an area where it's further apart in the same volume. So that's what's happening with diffusion. Now, this is a bit of a harder question. Explain the importance of maintaining a steep concentration gradient. Why is it important to do that, guys? So let's think. Why is it important? Well, steep concentration gradient. What does a steep concentration gradient mean? It basically means that the difference between the very concentrated area and the weakly concentrated area is a lot. It's very steep. And because it's a lot, what will then happen is it will allow diffusion to happen much more easily. So a steep concentration gradient allows that to happen very easily. And therefore, in order to write that in terms of GCSE terms, you can just say the greater the concentration gradient, then the faster the rate of diffusion. Because if we're looking at things that affect the rate of diffusion, we've got the concentration gradient as mentioned, but you've also got things such as the temperature as well. And then you've also got the surface area to volume ratio. So the concentration gradient, the temperature, and the surface area to volume ratio, each of these things affect the rate of diffusion. And that's why steep concentration gradient, therefore, so by maintaining a steep concentration gradient, you're allowing there to be a faster rate of diffusion. And so that with the same concentration, if it was the same concentration, then there'll be no diffusion taking place. If there was none of this steepness and just said here is the same as there, no diffusion will take place because there has to be a difference for diffusion to occur. Okay, and let's just kind of end with this last 6 marker now. Describe the adaptation for transport in cells. Now, this is a very broad question. It kind of summarizes that final GCSE cell biology topic. So what are the adaptations for transport in cells? Now, you can talk about a plethora of different cells here, and that's where the mark scheme is endless for this question. So you can talk about the cells in the small intestine, otherwise known as the villi. So you can talk about how they have microvilli, which is the even smaller finger-like projections on the finger-like projections. And what that does is it increases the surface area to volume ratio. Now remember, villi is actually where active transport takes place, which is kind of the opposite of diffusion. Diffusion is going from an area where it's really concentrated to an area where it's less, so therefore it's very passive, whereas active transport is going from an area where there's less to an area where there's greater particles. So imagine you're walking through a corridor, and diffusion is the person that when there's loads of people next to you, imagine there's loads of people next to you, diffusion is when you're moving from an area where there's loads of people in the corridor to an area where there's literally one other person there. That's diffusion. And it doesn't require much energy to do that. It's quite an easy process because you're going from an area where it's congested to an area where there's less people. Whereas active transport is entirely the opposite thing. So it's going from an area where there's less people to an area where there's loads of people. And trying to squish in between all of those people, that's going to require energy, whereas diffusion doesn't. So active transport requires energy, and therefore a useful adaptation for that would be having lots of mitochondria. And villi actually do. They have lots of mitochondria in the neighboring cells. So that's one example you can talk about, small intestine. Remember, they said plant and animal. So let's perhaps look at an example of a plant. So we can say root hair cells in plants. Now, one key thing is in their name, that they have a root hair cell, and this is a very long projection. And the benefit of it being a long projection is, as probably obvious, it increases the surface area to volume ratio. Another key thing is in root hair cells, it doesn't have chloroplasts, which again increases their area for you to be able to have to be able to intake more water. So therefore it's good osmosis rates. And then let's look at one more. So let's look at gills. Sorry guys, I'll come to your questions soon. But yes, just looking finally at gills. So gills are in fish filaments. So a key thing is that a lot of people forget about gills, but what it is they're just very thin parts of the fish which kind of just flap about in the water. It's kind of hard to draw. And the whole idea is what happens is the water goes through the gill and then into the fish. And then the fish then retains the oxygen and the rest of the water comes out. So what's happening here is a lot of people think that fish literally breathe using the water, but no, in reality, they don't breathe using the water, they breathe using the oxygen. They just use the gills to kind of let that oxygen be taken in. So gills are important. So that's how fish can drown. A lot of people think that's funny, but fish can drown, guys. All it needs is if the gills stop working, because they also need oxygen. So advantages of gills are the fact that it's very thin, which means that it's a short path of diffusion or osmosis. And also the fact that there's a lot of gills, it increases the surface area to volume ratio. So that will be it for today, guys. Thank you so much for watching. Just responding to your comments on YouTube, how do you recommend best revising? What do you think is best to do now? So in all honesty, my revision was very last minute, so it was pretty much from Easter forward. I went full on and I did a lot of GCSE English practice because that was my weakest topic. And I did a lot of GCSE English practice, such as just writing essays and writing loads of them, because I was predicted a grade 3, but managed to get 100% in the exam. So that just shows that even though I started at Easter for that, you can still make massive progress. So from now, I would say year 11s, the best idea is to, I would say even just half an hour of revision a day, unless you have mocks right now. Half an hour of revision a day is perfectly fine. And then Easter, I would say just ramp it up, because otherwise you'll get very bored of the content. Do you do lives for history, French, maths, GCSE, PE? So I don't do it for history and PE, but I will be doing it for French and maths. I failed GCSEs 5 times. I just want to go to university, but I'm not passing English GCSE. Girl, I feel you. I was struggling as well. I was also on a grade 3, and my teachers had no faith in me. I do GCSE English tuition if you really want that help, but I'm trying to help you guys a lot. I've posted a lot of my GCSE English essays that got 4 marks. I also post basically all of the resources I use. I'm trying to share with you guys as much as possible for free to give you guys a platform. So if you were to go on my website, which is one of the links in through my YouTube channel, you will hopefully see some documents there. But if you want some private help, then yeah, feel free to contact me. Okay. And yes, so I think that's all the questions unless anybody has any more. So well done, guys. You're definitely much more proactive than the previous year group. A lot of you are watching this and staying on. So really well done for that. Also, before you go, actually, there's a £50 giveaway that I'm doing. I just uploaded the TikTok on that. And all you have to do is subscribe and get other people to subscribe, and then just send me screenshots of everybody you got to subscribe along with yourself. Email it to me, and then how many people you got to subscribe. Say if you got 5 people to subscribe, then your name will be put in 5 times and you have a 5 times more likely chance of winning. And there'll be 3 prizes. So the first person gets £50 worth of things, and then the next two people get £10 gift cards. So make sure you go in those as well. The email will be nowafflelearning@gmail.com. Okay, guys, that's it. So make sure you join that giveaway and have a really lovely day. I'll see you tomorrow. Tomorrow I'll go through GCSE Physics Paper 1, and then the following day it'll be GCSE Chemistry Paper 1. And then just keep giving the recommendations, guys. So thank you so much for watching. Have a lovely day and see you soon. Oh, sorry, I know it's supposed to be in 2 hours. It just got cut short today because I'm a bit busy now. But tomorrow onwards it'll be 2 hours, so sorry about that. But yeah.